Dear students, today we are going to study phytochemistry and how this is important for taxonomy. That is, what is the role of phytochemicals in taxonomical studies and identification in modern times. Now, what is phytochemistry? The data from the chemistry of plant products is called phytochemistry. This is also called chemotaxonomy because we are discussing chemicals and their role in taxonomy, or phytochemical taxonomy. It is a study of chemical variation in various plants and utilizing this data in knowing their relationships. Such information is highly useful. One is for improved existing plant classification so that we can put those genera together in which same type of the phytochemicals are found. And secondly, this is to add to the knowledge of phylogeny or evolutionary relationships of taxa, because if they are having common ancestry, then they may be inheriting same type of the production of phytochemicals. So that forms the basis of its importance in taxonomy. Now, what are the techniques of chemical analysis in plants? There have been many techniques available nowadays to study chemical profiling of plants. There are different chemicals, like we may identify them at the genetic level, like DNA, proteins, enzymes, or there may be some compounds specially synthesized as a result of metabolism, which are called the secondary metabolites. Now, different techniques which are used for their identification or separation, they are chromatography, electrophoresis, protein sequencing, PAGE, RAPD, RFLP, PCR. All these techniques, they are highly sophisticated techniques which can give you an accurate information about different metabolites. Now, chemical constituents which have been employed in identification, they have been categorized under three headings. One is directly visible characters. You can identify, for example, the presence of raphides, the presence of silica. Second is secondary plant products, which are generally phenolics, terpenes. Third is DNA polymorphism and primary gene products analysis, means you can study the various types or the diversity of enzymes present in the plants. Now, first category, the directly visible characters. In this, we have raphides. What are raphides? These are needle-shaped crystals of calcium oxalate and calcium carbonate. They are characteristic of orchids, means the members of family Orchidaceae, and other families like Balsamiaceae, Rubiaceae, Saxifragaceae. On this basis, some decisions in the taxonomical classification have been taken in the past. For example, earlier Trappa and Monteva, they were included in family Onegeraceae. But due to absence of raphides, these genera have been replaced under Trapaceae. Because under the members of Onegeraceae, all of them, they are having raphides. But since both these plants, they were not having raphides, so they have been kept under Trapaceae. Similarly, Hydrangea, that was kept under family Hydrangeaceae and not Saxifragaceae, again because of the absence of raphides. The shape of crystals is highly important as a taxonomic marker because it may vary, and it has been used in the alien taxonomy where we have found the rectangular shaped raphides are not needle shaped. So here you can see the pictures. These are the raphides which are generally found. You will observe these in the members of the aquatic hydrophytes as well. And second, you can see these are needle shaped. And the next one in allium, the shape is totally different. They are rectangular. So they have played a very, very important role in the taxonomy of allium plants. Second observable character is presence of starch grains. Now, starch grains are also having variable shapes, so they can form the basis of taxonomy. They develop in plastids. They may be simple, compound, centric, or eccentric. Centric means when their hilum is present in the center and starch is deposited in the layers, and eccentric means when it is present at a displaced position offside and then the starch is laid in the form of layers in the eccentric manner. Then Titioka, he has delimited the tribes of Poaceae on the basis of these starch grains. Third character is silica. We all know that silica can be easily observed in the epidermal cells and members of some family, especially grasses, that is Gramineae, which is also called Poaceae. These play an important role in the taxonomy of grasses. Fourth is gypsum. It's a rare substance but present in the members of Taximericaceae, and here we observe the crystals of gypsum. Chemically, it is calcium sulfate dihydrate. It has been seen to be present in the members of Taximericaceae. Next is lapichol. Lapichol is a yellow colored powder which is chemically 2-hydroxy 3,1,4-naphthaquinone, and this has been observed in the wood cells of family Bignoniaceae, in the Avicennia of family Verbinaceae, and in Basia of family Sapotaceae. So it is highly characteristic to these groups only and not present in other members. This is the chemical structure of lapichol. So these were all easily observable characters which are morphologically visible and they can be differentiated. They can form the basis of classification. Second main category is secondary plant products. Secondary plant products means they are the result of the metabolic pathways of the plants, but they are not directly involved in energy production. Those who are directly involved in energy production, they are called primary metabolites like carbohydrates, fats, proteins, etc. But these compounds are not related to the energy synthesis, and mainly they are important part of the defense mechanism of the plants. So that's why they are called secondary plant products or metabolites. They can also be used as the chemotaxonomic markers in classification. The first such category is phenolics. Phenolics, most common type of the phenolic compounds are flavonoids. These are found in leaves, flowers, and fruits. They have been used in taxonomic studies like in bamboos and grasses, etc. On the basis of the profile of phenols present in these plants, they have been categorized. Second is betalanes. Betalanes are another type of the compounds which are having nitrogen in them. The broad categories of betalanes are betacyanins and betaxanthins, and they have been found in the members of order Caryophyllales and family Cactaceae. You can see here yellow colored compounds and red color compounds, they are present. So their profile can also serve in classification. Third is proteins. There are plenty of literature present in the books, in the research, where you can see that protein profiling has been used as a parameter for taxonomy. Like Johnson used this for the taxonomy of grasses. Vaughan used this for Brassica species. Free amino acids, these are generally characterized by chromatography, and they have served as markers for grass taxonomy. Last is oil, fats, and waxes. These are the molecules which form bulk of plant tissues. We all know that in plant tissues, the waxes, fats, and oils, they are present, and their profile, their composition, like fatty acids, whether they are unusual form of fatty acids present or their percentage of the saturated and unsaturated forms, etc., that can form the basis of taxonomic classification of different families. Now, next secondary metabolite is alkaloids. Alkaloids, they are highly restricted and present only in few genera in plants. Like lupin is found in Papaveraceae. Morphine is found in Papaver somniferum. Strychnine occurs in strychnos, so they are highly species specific, so they can be helpful in the identification. Similarly, Mears et al., he has also used alkaloid profile as a main criteria for classification of members of Fabaceae. Next category is cyanogenic compounds. Cyanogenic means in which carbon-nitrogen bond is present. These are called glycosides like amygdaline, prunacin. These are found in the members of some specific families only, like in Eraceae, in Juncaceae, and Gramineae. So they are a useful source of information for classification of these groups. Next is terpenoids and steroids. The Mirov has used the gum terpenes as the main classifying agent criteria for pinus species. These are mainly terpenes. These are categorized into monoterpenes, sesquiterpenes, diterpenes, depending upon the isoprene units they are having attached with them. And more are the isoprene units, lesser is the volatility of those compounds. So most of the compounds in which you find some kind of aroma or essential oil, it means they have abundance and a variety of terpenes present in them. And second compound here is steroid, that is also used for this purpose. So that was all where secondary metabolites they are used for taxonomic studies. Now the third one is the genetic material and primary genetic products. Here we have first is the main material, that is DNA. Itself, the DNA can be used as a molecular marker for taxonomical studies. In the recent techniques, we have used polymorphism in the DNA as the main tool for identification. And in this case, we require restriction enzymes and PCR technique for amplification of DNA. This method has been used in taxonomy of bamboos. Second is primary gene products, means enzymes. Now, various techniques like PAGE, that is polyacrylamide gel electrophoresis, then starch gel electrophoresis or isoelectric focusing. These are the techniques which have been used to study the profile of enzymes that are the first primary products which are formed after the translation from a gene. So they have been also been used in the taxonomic studies for many crop plants, and it has been seen that there are some issues with this, because you all know that genes they are under a regulation mechanism, and the enzyme profile according to the functioning of the gene, whether it is switched on or switched off at different stages of life cycles, the enzyme profile also keeps on changing with the developmental stages. So that is the one limitation which is associated with enzyme profiling. This is a just an outlook how the page is carried. Page is a kind of electrophoresis. You can see here we have a gel and it contains certain wells. In these wells, the specimen is loaded for which the profiling is to be done. It means this will be of species 1, 2, 3 and so on. We may use certain standards also. The gel is immersed in a buffer solution, and when the charge is applied, the movement of the molecules is from negative electrode to positive electrode. The smaller fragments, they travel farther distance and they reach the other end of the gel, whereas the larger fragments, they move slowly, so they remain close to the well. So this is how the separation that takes place, and later on these patrons, they are mashed for putting a particular species to different class or group. This is a restriction fragment length polymorphism, means we need restriction enzymes for this. The DNA is chopped with the help of restriction enzymes so that the fragments are generated. Then we run gel electrophoresis. Those fragments are transferred to a membrane, and that membrane is further treated with radioactive probes, and we obtain a bending pattern which is compared. Then PCR is meant for amplification. In PCR, we have a DNA sample. We use different primers, nucleotides, buffer, Taq polymerase. All these components, they are actually meant to increase or amplify a particular DNA sequence so that we have millions of copies of that DNA for further analysis. So this is polymerase chain reaction. So that was all that how different metabolites they are used for taxonomical studies. Now we are coming to the numerical taxonomy part. What is numerical taxonomy? When we studied different stages of taxonomy, then we had mentioned there that this is the most advanced technique which is being used nowadays, that is numerical taxonomy. It is called Taximetrics. It is a method of classifying organisms with the help of numerical means. We have different softwares in which we put in all information about the characters, whatever is known for that plant. And on the basis of that software, it is going to finally give us number of hits to which that a particular unidentified member should belong. So nowadays, the numerical taxonomy is very important in modern systematics, and its main aim is to classify organism using numerical algorithms. The period it started in the year of 1957 to 1961, when the first method of numerical taxonomy was developed, and the organisms they are arranged in their respective taxa on the basis of similarities as well as differences. So both are considered. Coming to the brief history of numerical taxonomy, Michael Edinson, who was a French botanist, he planned to assign numerical values to the similarities between organisms, and he also proposed equal weightage should be given to all characters. And these classification they were called after his name Edinsonian classifications. Later on, Sokal and Sneath, who contributed maximum to this type of taxonomy, they divided and they studied phonetics, in which classifications were based on the patterns of overall similarity, and cladistics, in which classifications are based on branching pattern of the estimated evolutionary history. So they divided this study, the pattern, into phonetics and cladistics. Phonetics, where we are discussing the similarities, overall pattern, what is there, how many characters are similar. And second is cladistics, in which we are going to relate that to the evolutionary history as well. So the numerical taxonomy was, however, largely developed by Sneath and Sokal. So major credit has been given to them because they somehow related morphological characters and the whatever data is obtained from other sciences to the phylogeny. The application of Edinsonian principles and the use of modern methods and electronic data processing techniques, that is the development of computer languages, it has helped the evolution of several new classifications of plants during the past few decades, and they are all the phylogenetic systems which are called APG systems. So what are the principles of numerical taxonomy? So whenever we have to use this particular branch of taxonomy for identification and classification, we have to follow certain set of rules. These are: greater the content of information of the taxa, more the characters are taken into consideration, better a classification system will be. So you can include as many characters as are known to you. Every character should be given equal weightage. Okay. Then for comparison purpose, the similarity should be considered. Correlation of character differs in groups of organisms under study. Thus, distinct taxa can be recognized. Phylogenetic conclusions can be drawn from the taxonomic structure of a group and from character correlation assuming some evolutionary mechanisms and pathways. So we can have phylogenetic information also to be included in this. And the science of taxonomy is viewed and practiced as an empirical science. Phonetic similarity is the basis of classification, which is related to their phenology, means their outlook, that is the morphological characters. So what is the procedure adopted for this? First is you have to construct the taxonomic groups. Then individuals for that are selected and their characters are spotted. Larger the number of characters, better will be the approach. The resemblance among the individuals is then established on the basis of their character analysis, which is usually done with the help of softwares. Then next is the discrimination of the taxonomic groups. So when the taxonomic groups chosen for the study, they show some overlapping of the characters, then discrimination should be used to select them. So there may be some characters like you may, let's suppose you started this analysis and you got a hit of around 10 genera to which your unidentified sample may belong. So then you have to follow the discrimination studies, that is the dissimilarities, so as to finalize what that genus is exactly. So characters which are taken into consideration are morphological, chemical, cultural, metabolic. You can have antigenic data, biochemical profile, physiological characters, or ecological characters. So here we are giving one example that how we have to find the matching coefficient. So you have organism A, this is organism A and organism B. So they are compared in terms of characters, they do and do not share, okay, which are similar and dissimilar. So the terms of association coefficients, they will be find out like this. So you have A, B, C and D. So the number of characters coded as present for both of the organisms. So you will write here if 1 means a particular character is present in both. How many such characters are there which are present in organism A as well as B? Let's suppose 10 characters are there which are common to both. So you will write here at as A. Then B and C, that is the number of characters which differ. So for example, B is the character which is present in A but not in B. So how many such characters? Let's suppose 3. Then similarly, vice versa, we are considering not present in A but present in B. So that will be considered as C. Let's suppose there are again 3 such characters. And D is the number of characters which are absent in both the organisms. So you have used that as a criteria of classification, but none of these A or B, they were having that character. Let's suppose there are 3 such characters again. Okay, so this is how you will have all these, this table prepared. So total number of characters will be just a summation of all these. So here that summation comes out to be 19. Now the simple matching coefficient, what is that? You will have A plus D. So 10 plus 3 upon summation of all of these, that will be 13 upon 19. So you will calculate that and this will give you the matching coefficient. A Jacquard coefficient, how that is calculated, that is purely on the basis of the characters which are present in both of those organisms. So that is 10 upon the sum total of those which are present in either of them or both of them. Okay, so that will account for 16. So this will give you the Jacquard coefficient. Here we are not considerate about the characters which are present in none of the organism under consideration. For example, let's suppose we have these 2 flowers. You are well aware this is rose, this is petunia. So we have simple leaves. In A we have compound leaves, we know. So here we will put 0, here 1. Stipules here are present, not present in petunia. Here the flower is solitary, here also that is solitary. Sepals here are free, sepals are free, they are not free means they are fused. Petals are free, so they are not free, fused. Stem is squarish, so it is not present in either of them. So this is how you come to know the characters which are present in both of them. Like this character is present in both of them. And similarly, this is the present, let's suppose the another one which is present only in A. So you will have this one is only in A, then another, this is only in A, this is only in A. The one which is present only in B. So this first character is only present in B. And the last is which are present in none of these. For example, this character is present in none of these. So you will have different values of A, B, C, D, and on that basis you can find out the coefficients. So after character analysis, the association coefficient that we just discussed, that is calculated. The association coefficient is a function that measures the agreement between the characters possessed by 2 organisms at a time. So simple matching coefficient and Jacquard coefficient, they are calculated. The simple matching coefficient, which is called SSM, that is a proportion of the characters that match regardless of whether the character is present or absent. So we have considered D category also in that. Whereas in the Jacquard coefficient, that is calculated by ignoring any characters which are absent in both the organisms. So we don't take into account D category in Jacquard coefficient. Both these coefficients, they increase linearly in their value from 0 to 1. So if there is 100% match between those 2 species, that will give you the overall value 1. And if there is no match between them, that will give you the value 0. So this is showing you we have my first bacterium, second bacterium. If you get this value, it means both these bacteria are exactly same. You may have some variations in that, and accordingly the value will decrease. So this is another example, the plot that you exactly get. After that, we are going to relate those, okay? Like 1 and 2. You can see here the values, they are similar, 1 and 2. The next is these may be related to 3 because again their values are close to that. This entire group, so you can see 4. 4 is totally different. The values are lesser. Then 5 and 6, they are more closely related. Their values are closer. So on the basis of it, we are calling it association coefficient. So obviously it is telling us how closely the species are related. So this is how ultimately we are going to plot their percent similarity. Okay, so this is how the softwares they work, how they perform in numerical taxonomy, and they give us the complex data processed in the form in a simpler form so that we finally identify the unknown species that we want to place under some group. Now, taximetrics in relation to taxonomy, these are the examples where it has been actually used, like delimitation of genera like Oryza, Sarcostema, Solanum, and many more. They have been separated on the basis of numerical taxonomy data. In the study of several other angiospermic genera like Epocinum, Chenopodium, Crotalaria, Cucurbita, Oenithera, etc., this has been used. Then phytochemical data from seed protein and mitochondrial DNA data from RFLP technique, they have been merged with the numerical data by Mondal et al. And have been studied for this identification of interspecific variations among 8 different species of Cassia. So here the taximetric has been overlapped with the biochemical data. Now, what are the advantages of numerical taxonomy? The data of conventional taxonomy, that is improved by numerical taxonomy, because we are taking into large number of characters at a time. Data obtained can be efficiently used in construction of better keys because we can identify the relationships. So we can accordingly assign different groups, which groups are to be closely placed, which are to be distantly placed. That all information can be generated from this. Then many existing biological concepts, they have been reinterpreted, that is just on the basis that earlier they were probably through the phylogeny we have come to know that those distantly placed groups under the natural systems, they are actually closely related. So this has happened in the past, because of which the APG system that has resulted in shuffling of lot of genera on the basis of their evolutionary ancestry. So this has helped a lot in reinterpretation. The numerical taxonomy allows more taxonomic work to be done by less highly skilled workers, because here now we are not that a single taxonomist has to attribute a lot of time in finding out, in designing a classification considering just 50 or 60 characters, but you can have n number of characters. You just have to feed that data in the software and it will directly give you the result. Of course, there is a need of an expert taxonomist, because characters should be properly identified first, and that is a part which cannot be performed with the help of a software, so or artificial intelligence. So this is really really important that expert taxonomists should first finalize the characters, their presence or absence, and afterwards the laborious task is done with the help of software. So this has actually made the taxonomic work very very simplified. Now, what are the disadvantages of numerical taxonomy? Numerical taxonomy methods are useful in phonetic classification, but not in phylogenetic classifications. It is generally because phylogenetic classifications are typically based upon the origin. Okay, the exact information about how these species they have originated, whether it is from a common ancestor or from the different lineages, that cannot be predicted on the basis of just comparing the characters. Then character selection is the greatest disadvantage in this approach. If the characters they are inadequate, because let's suppose I am having a list of 100 characters, and out of those 100 characters 50s they are not much significant. Okay, then but the statistical method is going to give all these characters equal weightage, then you may not get a satisfactory solution. Then different taxonometric procedures they may yield different results. Let's suppose you are having software A, some other data will be generated, software B that will give you some other kind of the results. So it is always going to give you different results. So difficulty is that which one is to be selected and which is to be ignored. So that really need a lot of experience and expertise. So these are the major disadvantages of numerical taxonomy. So with this, we end the topic of taximetric or numerical taxonomy and the phytochemicals, their role in taxonomy. Thank you.